show you the various paths in life science. Thank you. Thank you, one and all.
Thank you, ma'am, for your warm welcome. Now I request Srimati Sugandhi S. Singh, Associate Professor and Head of the Department of Physics, to introduce our guests to the program.
Good afternoon, one and all. It gives me immense pleasure to introduce our speaker of today's webinar, Dr. Navin Sharma GV, Head R&D, Nuzuvedu Seeds, Hyderabad, to this webinar. He has done his PhD from Kuvempu University, Karnataka, in collaboration with International Rice Research Institute, Philippines, and Beijing, China, with over 15 years of success in research and development, project management in biotech sector, with main focus on molecular markers, molecular breeding, trait development, biofuels, regulatory, GM, R&D infrastructure. His field of expertises are drought testing, precision phenotypic for drought field trials, end-to-end product development, project management, GM pre-commercial management of the projects, managing molecular biology and molecular genetics oriented R&D projects. He has published various papers. To name a few, he has published candidate genes for drought tolerance and improved productivity in rice in the Journal of Biosciences. He has also published SCAR marker for a QTL controlling maximum root length in rice. Dr. Navin Sharma has given this paper publication in Rice Genetic Newsletter. Key projects undertaken by Dr. Navin Sharma are: he has worked as research associate in Rockefeller funded projects on identifying QTL for root characteristics in rice. Then he has worked as a platform manager in genomics, head genomics also. Since time is the limit for the introduction, so I have given a brief introduction to Dr. Navin Sharma here. Thank you, one and all.
Thank you so much, ma'am, for the short and sweet introduction of our speaker today. Now I request Dr. M. Devika, ma'am, to present inaugural speech.
Thank you, Shakuntala. A very good afternoon to one and all. Hope all are safe and keeping good health during this pandemic. First of all, I'm really proud of our college placement and career guidance cell, as it is working in the direction of helping our students to get prepared for their future career and to get better placements. At the outset, I would like to express my heartfelt gratitude to Dr. B.M. Subraya, Sri N. Chandrasekhar, Honorable Secretary of Sarada Vilas Education Institutions, for their continuous support and encouragement in organizing this type of programs. Next, I would like to extend a very warm welcome to Dr. Navin Sharma, Research and Development Head, Nuzuvedu Seeds, Hyderabad, for accepting our invitation and is here amidst us to guide our students. Dr. Navin Sharma is a proud alumni of Sarada Vilas College. I really feel very proud to announce that he was a student of Sarada Vilas from 1993 to 96, and today you can see his achievements. He is well versed in executing molecular biology oriented research and development work and also in carrying out commercial projects. I request Dr. Navin to inspire our students in such a way that they should start thinking in a different line. They should start thinking seriously about their career in life sciences. Many of our students are really interested to get into research field, or they are really very eager to shape their career in a better way, but they are not familiar with the avenues. They are not familiar with the opportunities available, maybe due to lack of exposure. That is the reason why our placement cell has organized such webinars. I feel this type of webinars will definitely help our students to think in a different way and start shaping their career during their graduation itself. I express my heartfelt thanks to Dr. Naveen for having accepted our invitation and here today to help our students. I request all my students to make use of his expertise in the field, interact with him, and find answers to your queries or your doubts. Next, I would like to thank our technical team for their hard work behind organizing this type of virtual meetings. My special thanks are to placement officers of our college, Dr. Jagdish Krishna and Dr. Jyoti A.M. I thank all the faculty members of our college who have worked directly or indirectly in organizing this webinar. Thank you, one and all. Stay home, stay safe, and please make use of the vaccination drive, which is being launched in our college by the government of Karnataka. Thank you again. I wish all the success for this webinar. Thank you.
Thank you so much, ma'am, for this inaugural speech. Now I request today's speaker, Dr. Naveen Sharma, to take over the session. Over to you, sir.
Thank you, ma'am. Ma'am, Devika, ma'am, can you hear me?
Yes. Yes.
Okay, great. I do not have the visibility to ask each and every one, so that's good enough for me. Thank you for this organizing this wonderful session. It's for me, it's a proud moment that I'm coming back. I think, ma'am, Devika, ma'am, I think we. In 1995, you joined the college, right? So, if I am not mistaken,
No,
so we were in second BSc or what? 94.
92.
1992. Okay.
Yeah.
Okay. So, well, 1993, 1994, 95, 96. Did my BSc botany, biochemistry, and microbiology there. I think, if I'm not mistaken, I'm the first batch of student in that particular course.
Yes.
I think we were 18 of us, and then I think we were all curious. Some of us purposefully didn't want to go to engineering. We couldn't get the medical field, so it took purpose with so much of interest. We took this course, which was apart from Uvarajas at that time. This was the only college. Our college was the only one which was offering this course. Later, JSS, St. Philomena's, everyone started. But I think that time we were 1995, 1994, we were the only two colleges who were offering this course at that time. Remember that. But that was a new course. And then we were so eager about what to do, how to do with our limited resources. Learned quite a bit, actually. Helped a long way. The way it turned out is that we and Nagesh sir and Ma'am Yu and everyone who are so new, Namita, who are so new, we together kind of built this department up, especially the microbiology and especially the microbiology one. So it was really great 3 years spending in the college, and faculties were all really great, and that is continuing still. If you look at when I talk to Devika ma'am. It was really great for me to come back here on this forum and try to share my experience in terms of what I went through in terms of building the career from where we were in the BSc of those 3 years, then MSc, then PhD, all those things. So it was a great learning experience, and it is still. Want to share those instances with you guys. I will how I frame this going forward couple of around 20, 30, 40 minutes is that I'll not take you through the nitty-gritties of how the minute details of what needs to be done, where it needs to be done, and kind of stuff, because that becomes too huge. And also, I will not take you to one subject in depth. I'll try to justify the vastness of life sciences into a very concised manner. And if students are interested in any of those topics, I can always come back and spend some time and on an online platform and take in-depth in-depth course or in-depth webinar on that particular topic. I'll leave it to you guys to decide on that. Having said that, let me share my screen. Can you see my screen?
Yes, sir.
Okay. Great. I'll as I said, I'll try to do justice to the vastness of this life science, and try to touch upon the latest trends, what is going on in the field of life science. I'll not take you in depth with any of those things because it's too much. So before we start, apart from what is there in my CV, what Devika ma'am and Shakuntala ma'am read, it was had from 2000 onwards till now, almost around 21 years. It was a great learning experience in life. So it was right from BSc, it was MSc 2 years in Mansa Gangotri, biotechnology department, and then went on to work couple of years in an agriculture university, and then went on to do my PhD at Philippines International Rice Research Institute. Then came back, started working at a company called Avastajan in Bangalore, Avastajan Limited. I ended up heading the entire genomics facility for both humans and plants at Avastajan. After Avastajan, I went on to become Asia Pacific lead for a company called Syngenta. It's a world #2 seed company. There, my job was to basically, might have all heard about genetically modified organisms or GMOs. So my job was to make sure we commercialize GMOs in Asia Pacific, including China. So after that, recently past 2 years, I've been working on artificial intelligence in agriculture. and by using remote sensing data, satellite data, and other things, drone data, and other things. And also, I've been working as advising people on companies on cancer, on cell-free DNA assays, RNA assays for detecting smoking-induced lung cancer in the smoking population. This is just to name few, and then now I've taken up this assignment as head of R&D in one of the seed companies in at Hyderabad. So this is my brief portfolio. I'll take you through in detail in a couple of those pages so that all the students know how difficult or easy is to spend 20 years after BSc and look back and see, okay, there are kids who have just started doing their BScs in some of the life sciences, and then how far they need to go and what they needs to do. So having said that, see, life sciences is basically, we all know, it's basic animal, plants, and humans. So I'll not go into vertebrates, invertebrates, arthropods, and all those things. But just for the sake of having an understanding of what we are talking about, we look at animals, plants, and humans, which are kind of economically important, and also most of the research, cutthroat research or cutting edge research is happening in these fields. So basically, if you look at these things, so they are divided into three or four major subgroups. You can say health, diagnostics, upkeep, and genetics. So if you look at all these things, then you come back and look back on whether how human, animal, and plant would want to have health, diagnostics, upkeep, and genetics into their daily life, and what is the economical importance of that. Because I keep talking to you about economical importance and how much money is spent on all these things, because you will based on that, you will know which one is trending right now and what would be the major focus going forward in terms of in terms of the placements opening up, in terms of the field you would want to choose, and kind of stuff. When you look at things at human diagnostics and animal diagnostics, some of those things I have listed down here. So imagine a few years back, some of your family or colleagues or friends or acquaintances or elders might be going for scanning during their early pregnancies and kind of stuff, and then if there are any defects which were picked up in the fetus, they used to actually draw what they called as amniotic fluid from the womb, and then that would take almost around 21 to 25 days to get the results back, whether there are any defects in the chromosome, in the genome or not. So of late, 4 years, 5 years back, everything changed. So we don't need anymore an invasive technology, what they used to call as amniocentesis. But now they have the technology is advanced that can draw mother's blood and see whether there are any genetic defects in the baby, because something what is known as circulating DNA and RNA. So because that there is a balance between both mother and baby during the developmental phase in the womb. So there is a lot of exchange of genetic material, may it be DNA or RNA or protein or whatever it is. So between both mother and the baby. So the technology it has increased so much that they can pick up few strands of DNA and then do the sequencing of that and see whether there is any defect in those in the genome or not. So that is a kind of technology which has been in the forefront since almost around 4 or 5 years. Costs are gradually reducing. Earlier it used to be around 70,000-80,000 per assay. Now it is around 10 to 12,000 rupees. So there are these cell-free assays where you don't need to extract particular blood anymore. You just extract. You don't, for example, if you have cancer or somebody has cancer, a patient walks in with a cancer requirement. We don't need to do biopsies anymore with the cancer. We don't need to cut the cancer or whatever it is. Cancer tissues and then go and see the biopsies and see whether to determine whether they're cancers or not. Now there are cell-free assays where exactly the same free circulating DNA and RNAs can pick up and see whether there are these are any specific cancer types, where the organ of origin is, and all kind of stuff. So having to do all these things, there are separate companies who does all these in vitro diagnostics. So it's one of the major spending area where they basically create all these assays, these diagnostic companies. And that's one of the areas where they constantly are looking for people who have the zeal and who can work on some of these things. So I'll list out some of those companies going forward. You can understand. One of the major advances what has happened is in where there is ease of detecting something which is abnormal is imaging technology. Right. So, for example, most of you might have heard about angioplasty, right? So before that, they do angiogram and see whether there is any blockage in the arteries of the heart, and then they decide to put a stent or a bypass. Earlier, when almost around 15 years back, this technology was not matured. So that time, doctor used to give the chance of the patient to survive is around 40% survival rate, around 60%, 40 to 60 in terms of not surviving. But now, if you go and ask, my arteries are blocked. What are my survival rates? They without even a blink of the eye, they say 1%, 99% you will survive. 1% there are chances. This has been achieved because of the imaging technology. Without the CT, which they use to put the probe in to detect the blockage, and the ink to be able to detect the blockage, there wouldn't be any angioplasty or survival rates so high up. And then there are mammograms, there are MRIs, which gave a different meaning to the clinicians to be able to analyze the problem and be able to find the solution for it. And then there are implants. You might have heard about knee implants and all those things. Probably 15 years back, it was most of the implants used to fail. But now, because they use a lot of 3D modeling, 3D printing, and all those things, hardly anything fail unless there is a significant defect in the way it is done. So that's about diagnostics, and then there are medicine research where they seek for new formulations, new active ingredients in a lot of these disease segments like oncology, cancer, cardiovascular diseases, and the rare diseases. So that's one area where there is a lot of requirement for chemistry, a lot of requirement for people who are well versed with big data analysis, people who are good at genetics, people who are good at assays and sequencing and kind of stuff. So there are what we call in the next in the genetics, there are personalized medicine and precision diagnostics and medicine, where all these genetics requirement, which gene is mutated at what position and what can be done with those mutation, how to use those mutation for precision medicine thing. Generation, and there are a lot of personalized medicine which is required. So probably in a couple of weeks back or a week back, you might have heard about some very rare diseases, which is where that boy was the drug would cost around 13 crores or something. So earlier, if this was happened, nobody could have detected the disease and the boy would have perished. But now, because there is
Hello. Sir, your voice is not audible. Sir?
I think he left the group.
Madam Shakuntala,
Yes, ma'am.
can you call him?
Sure, ma'am.
hear me?
Yes, sir.
Hi, sir.
Okay. Sorry. It was I don't know what happened. Uh, Anyways, let's continue. How much time I have?
So 4:30, sir.
4:30. Okay. Not bad. Okay. So where we were? So we were at yeah, Decode and Me. So nowadays it is available in India, but we need to be very cautious in adapting those things because it's not been fully tested in India though. We are there. We are coming there because of the advent of all these low throughput sequencing and kind of stuff. So the next revolution, what happened during the course of the evolution of molecular research is the deciphering of human genome sequencing. So if there is any wonder which is happening in the field of medicine, which is way beyond what it was 10 years back, it's all because of the because we know what the human genome looks like, and we know even a single mutation what it means. So human genome is around 2.5 GB in length. You must all be knowing how the KBs and MBs and GBs are calculated from the nucleotide point of view. It's 2.5 GB, and when it started, it used to cost around 10 to 15 lakhs to go through one X coverage. One X means one time sequencing of human genome. Right now, for around 100x, that means that 100 times the coverage, 100 times the sequencing of 2.5 GB, it cost around 75,000 rupees or 80,000 rupees, and the cost will come down drastically still further. And there is no doubt people used to talk about $1,000 per genome. That is a past now. So there will be people started talking about $100 per genome, which basically is around at this rate it is around 7,500 rupees. So that has given large impetus into the precision medicine and personalized medicine aspect. And closely linked to that is an off-late gene. So CRISPR-Cas is a kind of a protein system where protein is
Sorry, sir, your voice is breaking. We are not audible it correctly.
and then goes and take a meeting. Oh, okay. ಆಗ್ತಾ ಇಲ್ವಾ? ನಂಗೆ ಮಾತಾಡ್ತಿರೋ ಕಾಣಿಸ್ತಿದ್ಯಾ? ಇಲ್ಲ.
Audio is not at all clear, Naveen.
Ma'am, is it okay now?
Now it's okay.
Now it's okay. Okay, I think
Yeah.
my I have a couple of Wi-Fi that by default it picks up some of the other network.
Okay.
Okay, so let me go back. Okay, let's some of the latest trends. Your personalized medicine and single gene target therapies using CRISPR-Cas. For example, the CRISPR-Cas is used to you remove a single nucleotide polymorphism which is affecting the which is causing the disease and replacing that with the other disease, the wild type ones which are not causing the disease. So in that sense, the technology per se is very much matured. But having said that, there are ethical issues which comes into foray. And things like that. So that we will talk about that later or some other time because it's too much. So when it comes to a plant, obviously there are the same things, but one advantage in the plants is that we can there are no too much of an ethical issue unless you're talking about genetically modified crop or plant. So there are a lot of feedbacks or setbacks in terms of GM technology. You must be all aware of that in the Indian scenario. So the scientific fraternity is still fighting on that with the NGOs and with the public at large in terms of managing the bandwidth of what is acceptable and what is not from the point of view of applications and kind of stuff. Because from the GM technology, what we are doing is we are taking out a particular gene which is not present in the plant system. And introducing that gene into the plant system, which gives a different tolerance levels for any particular diseases or pest or anything. So you might have seen pests eating your plants, or if you're an agriculturist, you know what does that mean. For example, cotton. In India, India introduced GM technology in cotton somewhere in 1994, 1993, 1994. So from that point, 2003, 2004. So from that point, can you hear me?
Yes, sir, it is audible.
It is audible.
So from that point, earlier when they introduced in 2003, it was around 2% or 3% of the entire cotton growing area, which was GM. So within a matter of 3 years, the the cotton, the GM cotton reached almost around 65 to 70% of the total cotton grown area. So what it did was that India became #1 cotton producer in the world, surpassing China. So that means quite a lot to different people at different level. So and it's always so important to be self-reliant in any of these, sustainably self-reliant in any of these areas. So having said that, there after cotton, particular government, because of so many issues and push from the NGOs and other things, we didn't take it to any other crop system. But from the point of view of science, it's a very well established technology. And there are a lot of opportunities there in terms of regulatory science, in terms of product safety science, in terms of generating the plants itself through transformation, and in terms of commercialization. There are a lot of opportunities there if you want to focus on those lines. Having said that, let's see what it means to a particular person who is aspiring to be, wanting to be in any of these fields. So let's take up humans and animals first, or human systems first. So this is very important. You always people who are aspiring to be in some of these fields needs to look at how the financials looks like, in that particular field. So, for example, medicine research, that means that identifying the target gene, target protein, trying to develop active ingredient to that, and all those things. It's just the public spend, it's the government spend. It's just in US alone. Forget about rest of the other countries. In 2017, it is $71.4 billion. Okay, 1 billion is 1,000 million, and 1 million means $10 lakh. So that is how the maths work. So 1 million is around at this rate it is around 7.5 crores. We are talking about 7,100 crores as 1 billion dollars. So that is the kind of money what they are spending on public research. So this is mainly spent on oncology, community health, vaccines, eradication program, large grant, pediatric science, those kind of things where the private guys are not very active. Okay. So that is where the focus of public spend will be. So next, we have large players in device. Some of these sequencers, some of these diagonal. You might have seen, if you have ventured into one of these path labs, there are big machines doing a lot of crazy stuffs. For example, earlier when we are looking at 15 years back, when we give our blood sample in the morning, we used to get 2 days later. Now the technology is improved so much, so we if it is required, we will get it in half an hour. So that is basically will be really useful when we look at the throughput and countries like India, where the population is so high, and then that is the kind of money what they put in. For example, the 2020 spent for just on the device by the private companies, and these are not government spends. These are around $49 billion. It's not huge. It's not small. and these are just private players. They don't have government backup. They're not having anything, so they just do instrumentation. They standardize assays and sell it to labs and research grants, and also universities and kind of stuff. So when 10 years back, when we were talking about sequencing, India had just around 4 or 5 sequencers to do any kind of sequencing activities in the next generation sequencing. So right now, every lab has one. Every university has one. For example, in Gangotri, if you go to your molecular genetics in zoology, they have one. Dr. Ramchandra has one probably, and the biotechnology must be having one. So this is how the penetration of our technology is inevitable, and there's a lot of research which goes on to that. And some of these are, as we spoke earlier, imaging technologies, diagnostics throughput, as we were talking about, sequencing, chemistry of diagnostics, and some of the new generation technologies like NIPT, cell-free assays, and kind of stuff. And the third one, which only the private money goes into, is in vitro diagnostics. This market is around 51 billion dollars. These are the spend which is happening per year, not through the years. And then overall, the human genetics market is around $1 trillion. So somewhere around 995 or 996 billion in 2021, at least, and probably because of this corona, might have surpassed $1 trillion long back. So again, these in vitro diagnostic market is all assays, cell-free sequencing, single nucleotide, single cell-free sequencing, and sequencing-based assays. And nowadays, there is rise of artificial intelligence-based analysis. So it's no more one gene, one mutation, that kind of thing. So if you the way it is done is that there are the chips, which basically there are multiple genes on the chips, which are printed, and then a minimum chip will have around 2,000 to 3,000 genes and multiple SNPs on those genes. So if you look at that analysis, there will be almost around 100,000 to 200,000 data points which you'll get. So it is no more put it in an Excel sheet and then see whether that mutation is there, this mutation is there. No. So all these assays are basically driven by artificial intelligence based assays. They take multiple gene models, multiple SNP models, and then put them together and get the scores of what it needs to be. Right. So and it has helped immensely in especially in the cancer research, and also some of the rare hereditary diseases. So if we go into next slide, so where all this money gets spent, this would be a really important for the starters who have just started the life science career or the academics, and then want to take it forward in terms of their interest and kind of stuff. So this is the 2021 trend.
Excuse me, sir. Sir, please share your presentation screen, sir.
It's gone it again.
Yes, sir.
Happened. Can you see?
No, sir.
No. Yeah. Okay. This is how it is. Okay. Hope you're seeing it.
Yes, sir, it is visible.
Okay. Just shout out if you lose it again, right?
Okay, sir.
So then this is really important. You need to really get a hang of where the future science will take us, because uh, when we when I was doing my MSc, we had to do a small 6 months, 3 months project work. You might have known this. There is the sericulture CSR and TI here. There is another branch in Bangalore which is dedicated to these silkworm genetics. You know how we used to do sequencing. This was in 1998, if I'm not mistaken. Yeah. So we used to tag radioactivity to a particular primer. Hope you all understand what I'm talking about: primer sequencing and kind of PCR and kind of stuff. So the radioactive tags to the primers, and then do the sequencing of that. So then there used to be a very long gel, almost around 2.5 feet, a very thin micron level gel, electrophoresis, which we used to run these nucleotide sequencing on, and then we used to dry it overnight, that particular gel, and then put a radioactive film in the dark and expose it for another one day. All these things used to happen in dark. So maximum thing what we could do was around 400 600 bases per day, not more than that. And then we used to put the scale horizontally and then say ATGC and kind of stuff. So it all changed drastically. Now we are talking about 100x GBs in 1.5 hours. So that is where the technology took us from 1998 to till now. And it didn't come easy. There was a lot of money spent. There was a lot of mistakes happened, and that's part of the game. So anybody who is aspiring to look at where the science is taking the future direction, they all need to look at where the money is spent right now. So what all I said about how much money was allocated or how much money spent, all those money was spent on these. So this is still half of 2021, and they have spent quite a big amount of money in these startups. So gene editing, precision medicine, gene sequencing, big data, artificial intelligence, biomanufacturing, synthetic biology. Synthetic biology is changing like crazy. So they're able to synthesize small bacteria without having to go through the whole biological process. So that is where the technology is going, and bioprinting. So probably when you take up any of these PhDs or some of the industries, there will be no more organ harvesting through a dead body or cadaver or that kind of system. So it's just they take up the DNA of an individual and then simply print the organs. So that is the future where it is heading. Example, tissue engineering and with bioprinting put together, it changes the whole dynamics of how the science, how the application point of view will be seen in 5 years down the lane, or 6 years down the lane, or 10 years down the lane. When when we started our BSc and MSc, right, especially me, my kind of age people, things were still laid back. Things were still slow. The research was not happening at a very significant pace. So imagine from 1998, when we started doing our sequencing using radioactivity, and the first draft genome, it took almost around human genome. It took almost around 10 years. By 2008 or 2009, we had a complete draft human genome, and then after that, we can do, as I said, 100x genome in 1.5 hours, right? So that is the kind of pace which is going on right now, and because of that big data which comes up, so it is no more ATGC handwritten. It is all big data, right? So it is all big data. So there is huge amount of bioinformatics work required. So it's no more a challenge to generate data. It's a challenge how we analyze it. So if people are interested, analysis part of statistics and analysis part is a major goal. If you want to pursue that way, that is one of the careers what we can look at. So okay, so I'll stop here on the biotech. So as I said earlier, I am not going to tell you what is PCR, what is sequencing, what is tissue engineering. No. So we can get back again after a few days. Talk to your officers, you talk to your teachers, collectively tell what is required. I can take you in depth of what each one of these are. But this is really important for you to be able to understand what is happening right now in the field of both human space and the plant space. Let's jump into plants. Okay, the plant is a separate entity. So we are not looking at a human life here. We are looking at feeding the world, right? There is a complete shift of thought process in terms of trying to design a drug for a personalized medicine, design imaging system for detecting the disease or kind of stuff. We are looking at how to increase the productivity of any particular crop. So, for example, corn, maize, right? You have all eaten popcorn or corn. You might have seen corn growing in the field, maize growing in the field. In 1940, or forget about 1940, that's when the first hybrid came in for the corn. So even as late as 1980, the average per acre yield of corn was somewhere around 2 tons per acre. So from 1980s to 2000 and 2001, 2021, so from 2 tons per acre to almost around 7 tons per acre. Okay, is the gain which has happened in terms of yield. Okay, it's the same. There are tenfold increase in the yield, in the productivity, in almost all the crops. So that is significant. Tenfold in 20 years is basically 5% increase in the productivity rate. That is quite significant. Right now, from past 3 or 4 years, it is kind of stagnated. We are kind of looking at a completely different paradigm shift in terms of how we do the research of gaining the productivity. and also use a different system. System of change in the genetic gain, which is pretty relevant for us to take these things forward, because how the population growth is happening. It's phenomenal, and there are around 7 to 8 billion people in the world right now, and half of them are in Asia. And feeding them is a humongous task, and the population will not stop. They will grow. So that is the challenge. The technology is basically remains the same in terms of sequencing, in terms of thing. The challenge here is that we have some kind of liberty to play around in terms of in terms of the plants. So, for example, here there are companies like Bayer, DuPont, Monsanto, Syngenta. A lot of these have gone into different changes of late. So there no more Monsanto, there no more DuPont. So there is Syngenta, which is a Chinese company now, which is not Swiss. Monsanto has been merged with Bayer, and now it's called as Bayer. DuPont and Pioneer is again came together and is called as Corteva. So from the point of view of all these changes, you can look at how much spending they are doing per year. So it's almost around $1 billion. That too in 2014. When I was in Syngenta till 2019. As I said, I was holding the APAC Asia Pacific lead position for genetically modified foods. So their entire spend was almost around $1.6 billion for per year. So that is the trend. So which is these things are categorized into 3 or 4 buckets, like farm machinery, as you say. These Deere, you might have seen John Deere tractor going here and there. So they're quite significant. CNH also is quite significant. Kubota from Japan is one of the leading people in tractor and machinery. Likewise, animal genetics and nutrition is one of the major things, because you need cattle and livestock and agriculture go hand in hand. And crops, basically, they are crops mean seed, chemicals, irrigation, and agronomy. Seed is one of the main things. So if you have seeds, it doesn't end up there. So there will be diseases which will come in. There will be, there will be damages because mechanical or physical damages, and irrigation is required, and agronomy is really important. So having said that, this is what goes into developing a particular hybrid. Which is kind of quite elaborate. So for every hybrid, for example, you see Sona Masuri, you simply buy it in the shop and eat it. For example, tur dal, togri bele, so kadle bele, green gram, black gram, urad dal, all these things. So it takes minimum of 5 years to be able to bring it onto a field where it can be tested, onto a farmer field. It takes 5 years, and onto your plate, it takes almost around 8 to 9 years. And each one of them has to go through. These are the very recent trends of how the plant breeding works. This is all what it takes to go to a particular from where it starts in the lab to the plate. It takes all these things, and one variety to be released So it cost a company almost around 12 to 13 million dollars. Okay, 12 to 13 million dollars per variety to be released, because it's like earlier, it's no more just go cross one female with male and then go to the other field, come back later and do the breeding. So it is not. Nowadays the competition is so high. So right from phenotyping, which is a mechanized phenotyping right now. What there is a separate subject called phenomics, where it's only talks about how do we phenotype a particular plant or a particular cropping system. And there is a lot of AI, robotics, everything is involved in there. So then there is genotyping, which is basically a lot of PCR markers, a lot of sequencing, a lot of SNP arrays, and genotyping by sequencing, and a lot of these jargons, a lot of these assays. And then there is a huge breeding strategies like marker assisted breeding and traditional breeding and kind of stuff. And then there is from how do we cut down from 8 years to 4 years. So all these things takes humongous amount of money. And as I said, it takes 10 to 12 million dollars and 4 to 5 solid years of research to be able to release a particular variety so that it can be on our plates. So the opportunities here are basically genetics, statistics, phenotyping, and it will help if it's an agriculture graduate. There is no harm in not having an agriculture degree. Not all agriculture graduates are well versed in genotyping and generation advance and phenotyping and kind of stuff. Because I am not an agriculture graduate, but still I hold a very high position in seed industry. So having said that, some of these are really well worth your life spent on, and the prospects of growing together with these fields are quite immense. So having said that, I'll stop with both plant and breeding recent trends there. And as I said, if you need any particular subject in particular, want to be lectured in depth, so let me know through Devika ma'am or others. So I can spend another 1.5 hours some later days and then take you through all these things. So let's briefly touch upon these opportunities. I believe you would have got a very broader view of what I was talking about in terms of where the opportunity lies, how to pick up these opportunities early on, and what it means to be a greater good to everybody else. So it's a no-brainer to show this, but still I want you to look at these and job roles, and don't see this as a job role. Don't see this as there are jobs like that. I want to simply pick it up. No, don't do that. So that is my experience. So look at where you want to be, which industry, what is your persona tells you, how to make it happen and kind of stuff. So then there are, if you look at a microbiology degree, so it is basically start your 3 years, BSc. At the end of the 3 years, you get BSc degree. You can as well pick up a job. There's so many people, my friends itself who have done BSc, they have straight away went and picked up the job. There's no harm in that. It depends what a particular person wants. Then there are a lot of entrance exams nowadays after the BSc, right from your central university exams to individual JNU exams to IAC exams. There are 101 exams, so you can pick and choose any one of them. Start early, be very specific what you're looking for, and spend good amount of time doing that. Rather make it as fun. Don't toil it as, oh no, I need to prepare for the exams. It's all to be understood. There is nothing which you need to toil here for. That's my advice. And then there is a PhD in microbiology. Once you finish, if you have that time, and then if you have the bandwidth to go through the rigor of PhD, just go ahead and do that. Take your time. It's okay. You do not have to continuously do your PhD right after MSc, BSc, PhD. No, not like that. If you want to take a few jobs, take it easy, and then if you feel like doing PhD later, yeah, go ahead. There's a lot of things you can do, and then there is postdoc later after PhD. Well, it might be relevant or it may not be relevant, but yeah, it's nice to spend certain time doing other than the PhD, what you have into some other similar subjects and kind of stuff. So don't think that these are the only job roles. There is nothing like a defined job roles. You can take up anything and everything. There's a lot of opportunities in the world right now. So this is, I'm kind of fathomed with this. I'm kind of intrigued by the way this has been built. Some of these are really, people have put in a lot of effort into building this particular tree. So it's kind of self-explanatory. There is, if you go on to the hierarchical thing, they feel that your the opportunities which is as a job or as a livelihood, because of that will improve. I have seen people who have grown with just basic degree, BSc degree, till the top CEOs and kind of stuff. And they have started their own company just by basic degree and employed postdocs. There is no end to it. Don't be disheartened, or don't be don't drift away. That's what it is. So just be focused. Just make sure that you're meeting your self goals, and then take it forward. So it's basically, the world is yours. Once you have a very strong basic foundation, what your teachers are giving you right now, after that it is up to us, up to you. From MSc onwards, nobody will come and tell you what to do and what not to do after BSc. So should be fortunate enough that there are people, there are teachers who are going to tell, you will regret later if you don't do it right now. So this is our experience with the way the conversation used to happen in the classes, where some of us are a bit naughty during the BScs as such. So if you reminiscence on that, so it is a learning experience. So the only thing is that just don't drift away. So there's a lot of opportunities. Have a strong footing. Learn quite a bit. Don't be scared of anything. Everything is achievable, and get the life going. There is no set rule for, okay, this is how you should grow or how you shouldn't grow. Anything is possible. That is how we shaped our life. So anything is possible. So let me take you through my course of how we grow. This is my last slide, probably on time. So I did my BSc. I didn't get for a medical entrance. I didn't get the score. I couldn't go. Got engineering. At that time, it was really tough, even to get engineering. So we used to be really good at ranking. So anyway, so that is something which I didn't want to. So I was looking for a life science. So then Mysore was our hometown, and then Sarada Vilas pretty well. Went into started doing this BSc in botany, biochemistry, microbiology. As I said, ours was the first batch. Kind of built the labs, learned by errors, learned by mistakes, good teachers, and I don't think so, other than Devika ma'am and Manjunath sir right now, anybody whom you know is there from our batch. So then did MSc biotechnology post graduation. Then after post graduation, at that particular point of time, what to do, right? So struggled for a year, hopping from a year to year, that organization to this organization with a CV in hand, a one-pager CV, which doesn't say much other than the project work what we did. It was an experience. So I think everybody goes through that. It's not a big deal at that time. So then started working after a year as a research associate. Set up a entire lab at that particular point of time in Agriculture University, and then went on to do a PhD from 2001 to 2006 in Philippines and China. Got a Rockefeller Foundation fellowship there, and then came back to Bangalore, worked at Avastajan as head of genomics. And then 2012 to 2019, I did my stint at Syngenta as an APAC lead. Then said, okay, let's look into enough of these structured job. Let's look into some of these advisory and consultancies. So we said, okay, we have. I'm working for 2 or 3 companies, including Robert Bosch, Avastajan, and the other what we call as Genomatics. Genomatics is an AI-based, AI-based analysis platform. Bosch is an again AI-based agriculture advisory for crop monitoring. These things, and now people are forcing again to go back and take up this head R&D for Mosquito seeds in few weeks. So when you look at things, I think it's an experience built on our own. So we are our own advisors, and we got into the right position and kind of stuff. And then there was teachers who contributed quite a lot in shaping things up. And basically, it was time worth spent. If you try to if you don't drift away and then just be on your own and be adamant on what you want to do. So it's a good time. It would be a good time. So in this phase between after graduation and after post graduation, if you if that particular point of time where you finish graduation and then you don't want to do post graduation, you don't know what to do, you can ask through your mams, teachers, and this is my mail ID. And if anybody wants it, and obviously you can ask me anything related to science, life science, and kind of stuff. So this is what I had to tell. Hope I am on time.
Thank you so much, sir. It was a very informative and useful session for us. The students are much enlightened with your talk. Now the session is open for queries. Any questions or clarification in this topic? Participants can post it in the chat box. Otherwise, you can unmute yourself and ask the speaker. Any questions?
Naveen, Naveen, can you elaborate a little more about the exams which they can take up after their graduation?
Graduations are in India. There's a lot of opportunities, right from the central exams which they conduct for life sciences, where there are around 24 universities where they combine together and do one particular exam. They can pick and choose the universities what they do, like for example JNU and IMTech and Pondicherry University and Madurai Kamraj University, Madras University, and kind of stuff. So there is this central network which goes in. Apart from that, there are individual exams like IISc and other big renowned. JNU also has their own exams, and then there is this integrated course in NCCS Pune, National Center for Cell Sciences. The IISc also has an integrated MSc PhD program, which is of 5 to 6 years right after BSc. There are quite a large pool of universities which offer great lengths. And also, if you write GRE, if you're interested to go out, there are integrated courses in in both MSc and PhD. So that would be an worthwhile thing to look into. If you're interested in doing both MSc and PhD. So the opportunities are quite big, but you need to write entrance exam for all these things, and you need to prepare. The competition is quite severe, quite huge. You need to prepare for almost around 6 months to 7 months to be able to even have a good chance to be able to qualify for that. So these are some of the areas where you can put your thoughts into. And job-wise, I kind of don't suggest to pick up a job right now with just with BSc. But in case if you need to, because for a lot of obligations, so it would be worth a lot of opportunities are there, like lab technicians and pharma companies are looking for marketing people every year. You can just go walk in and get the job, and pay is not small. They're quite huge. So from that point of view, I think it should be. Whether it is academics or whether it is job, there shouldn't be too much of a problem after finishing this, either the pure science, botany, zoology, biochemistry, chemistry, or through our BBMs and other things, biotechnology and other things. It should be pretty neat from the point of view of getting the job and academics either.
Any other participants have questions?
Yes, ma'am, I have.
Yes, you can ask.
Good afternoon, sir.
Go ahead, Swati.
Sir, after MSc Geology, is there any job-oriented courses are there that is short-term courses?
Job oriented. We'll consider zoology as a pure science. It would be there are a lot of short term diplomas which will give us some insight into either the biotechnology or the biochemistry. For example, Jain University, and nowadays there's a lot of other universities have short-term courses, uh, uh, either in person or through contact program, around a year's program, I think, where they will be given a diploma in biotechnology or biochemistry right after your pure science. But I think having the pure science knowledge, and right now the course is so dynamic that it doesn't matter whether you are a biotechnologist or a zoologist or whatever it is. I think it should be fairly, I can say, all of those who comes out as a master degree will have a same kind of knowledge in most of these fields, which is relevant to get a job.
Okay, sir, thank you.
Any more questions?
Sir, I have a question.
Go ahead, Rikshita.
Sir, like nowadays we know that the farmers facing a lot of problems related to the seeds, and I would like to raise one problem, which is seed germination. Even farmers buy the seeds from a certified place or a certified institute, or like other like seeds ranks. They are also facing a lot of problems like seed germination. What do you suggest to overcome this problem instantly?
There are a couple of issues. It's a deep-rooted issue, Rakshita. So the germination problem is not because of the seed per se. It is because of the company who is selling it. So, because when you look at the very established companies like Monsanto, Syngenta, and other big companies, in my tenure in the seed industry, I have not come across that big a problem which is persistent across the years. So being a farmer myself, what I have seen is that, for example, if you want to grow rice, that to a locally taken rice or ragi or preserved ragi, or go to a APMC or some other thing and get the rice. For example, if you want to grow an IR6 for a Sona Masuri rice, you need to have around 100 kgs of 150 kgs of rice seed to be able to have around 40-50% germination. That too, if you are lucky. But if you go to and buy seed from companies like these big companies, the germination rate will always be around 95-96%. If not, you can sue them double time, triple time, and they give you, without asking questions, they give you money. Or any other kind of thing. So it's kind of very well established norm in the industry that there are quality issues in some of these companies, and the issue is so rampant that even the agriculture department has stopped looking into that. My suggestion is to go buy seeds from these companies who have a truthful label behind. What we call as a truthful label on the back of the seed packet, where the germination is pretty well established.
Okay, sir. Thank you.
Go ahead, Rakshita.
Sir, I have a question. Sir, like nowadays we know that the farmers facing a lot of problems related to the seeds, and I would like to raise one problem, which is
Thank you, sir.
Thank you, Rakshita, for your question. Any other participants have questions?
So I have a question.
If no more questions. Yes, sir.
Regarding all those things Rakshita has said, like germination and all those problems, as we are student of basic science, and how we can look into it, and by not doing that much of research, how can we look into that problem and solve in our level? Even all the agriculture, means solution for doing solutions, we don't have any PhD or master degree. How we can look into that perspective of doing solving problems and also.
No, you don't need to have master's degree or PhDs to solve the problem for these kind of problems. So see, there are two things which I want to say. So when there is a truthful label behind the back of the seed packet, that is what the company is saying, is that they are abiding by that, even though it is they cannot lawfully claim it, because the Indian seed law doesn't permit any kind of legal proceedings against the company, because this label is not judicially bound. That's why it's called as truthful label. So when I say that, if the company has that, if the truthful label is there, you can go to an agriculture department nearby your village or taluka, and get this verified, and make sure that truthful label is abided for. And the second thing is, when we look at things in terms of what we can do without having a master degree and a PhD. So nowadays, government is encouraging any graduation, preferably the life science graduation, irrespective of whether they have agriculture graduates or not. For example, zoology, botany, or whatever it is, they can apply for what they call as plant doctors. So in every village or any taluka or any hobli level, there are government will give permission to establish these clinics. So what they call as plant doctor, or something like that, if I'm not mistaken. So it's closely, they work closely with the agriculture department, whether it's an agriculture or horticulture department, enabling farmers to be able to give these solutions, whether it is a pest or soil deficiency, or some of these things what we are talking about, seed germination and kind of stuff. So that is kind of, that particular person is allowed to get something from farmers at the end of the day, if that is how the revenue model works. So there is a nominal fee which is prescribed, and people are doing it well. What I have seen around Karnataka and elsewhere.
Any other questions? If no more questions, we can conclude the session. Now I request Dr. Jyoti, Assistant Professor and Head of the Department of Commerce and Business Administration, to present vote of thanks.
Thank you, Shakuntala Madam. Om Saraswatiyai Namaha. Good evening, everyone. On behalf of Sarada Vilas College, Mysore, I, Dr. Jyoti A.N., HOD and Assistant Professor, Department of Commerce and Business Administration, I feel proud to propose vote of thanks to those who have directly and indirectly contributed to this webinar on recent trends and opportunities in life sciences, organized by Placement Cell, Sarada Vilas College. At the outset, I thank our speaker of the day, Dr. Naveen Sharma, Head Research and Development, Nuziveedu, Hyderabad, for accepting our invitation and sharing many opportunities in life sciences to our young minds. Thank you, sir.
Thank you, ma'am.
I would like to extend my heartiest thanks to Dr. B.M. Subraya, President Sarada Vilas Educational Institutions, Mysore, who always encourage us in all our value-added activities in the college. Thank you, sir. I would like to thank Mr. N. Chandrasekhar, Honorable Secretary, Sarada Vilas Educational Institutions, Mysuru, for motivating us and giving us a moral support. Thank you, sir. I extend my thanks to Dr. M. Devika, Principal, Sarada Vilas College, for her enthusiastic support for organizing such a webinar. Thank you, ma'am. I extend my thanks to all heads of the department, Sarada Vilas College, for their untiring support. Thank you all. I would like to thank all our teaching and non-teaching staff for their excellent coordination. Thank you all. My heartfelt thanks to all our participants who have turned up in such great numbers to make this webinar a grand success. Thank you all. With these warm words, once again, I thank you all for your cordial cooperation. Thank you.
Thank you, Jyoti ma'am. The link for the feedback form will be shared in the chat box and to WhatsApp group. I hope this session was very informative. Improved technologies facilitates a lot of job opportunities in the field of life science like genetics, medicine, and agriculture. So enhance your ideas in the path of life science and enhance your career.